So what I will talk about today is inflammation and depression, and I will try to give you a rather broad perspective on it, rather than going into a lot of experimental detail. Now, if you came based on the flyer that Professor Cosentino spread, on which I guess there was a lot, and you realize that I may not be talking about some of the items that were in the flyer, please feel free to bring it up and I'll be happy to talk about it. I just thought that for a topic that maybe you're somewhat less familiar with, it was perhaps a good idea to start with a broader perspective with some context. And also, we're not that many. If something is not clear along the way, please feel free to interrupt. Okay, with this being said, so I have given somewhat of a cryptic title, which is in part on purpose. In part, it was also to gain some time about really what I was going to talk about. So inflammation and depression is nowadays kind of an interesting and link that's been made more and more. It has not always been so clear that the two were related, so I will also try to give a little bit of historical background. But as Professor Cosentino clearly said, my own interest and research has been mostly on the effects of inflammation and behavior. So my seminar today will be divided in 3 parts, not equal parts, but these points will at least be addressed. The first point will be to provide you with a little bit of background on the notion of inflammation, and in particular, whether or not it should be considered something that is like a functional response of the host to infection or injury, or whether it is always a maladaptive response. This is still an ongoing debate. I don't pretend that we'll close the debate today, but I will just give you an overview of the positions that have been adopted over time. Then I will spend probably quite some time on the notion of sickness behavior, which is a notion we have developed in Bordeaux to describe the behavioral changes that one can observe in our case, animals, but that we think are also relevant for human beings that are sick, most oftentimes from infectious diseases, but I think it can also be expanded to people suffering from tumors. And so we'll talk a little bit more about what this sickness behavior is and some of the mechanisms that may be underlying some of these behavioral changes. And my last part will be about the question of whether inflammation and depression, how to approach this question. I will spend some time on detailing a little bit the different notions of depression, from a word that was just a word in English to something that became a clinical entity. And I will talk a little bit about the possible involvement of pro-inflammatory cytokines in depression. So to start with the first point, inflammation, functional or dysfunction. So Virchow in the 19th century very much promoted the idea of inflammation being something dysfunctional. Up until then, at least for local inflammation, we had the 4 classic symptoms that were already described by the ancient Greeks. And Virchow very much emphasized that we should add a 5th symptom to those classical symptoms, which is actually the loss of function. So he very much prone an idea of inflammation as being something dysfunctional. So that gave us these kind of cartoons with the 4 classic symptoms of local inflammation: the heat, redness, swelling, and pain, all local. And then Virchow added to that loss of function. Now, how does that translate to this general idea? How does it translate to specialized cells? He makes the case that an inflamed muscle would not perform its normal role, and the same thing for nervous cells. For him, it is straightforward that nervous cells being exposed to inflammation would not function normally and not respond in a normal way to stimuli. So that is one extreme position, but I think it is, at least it has the merit of very clearly pointing out what one of the positions in this debate is. Addison, a famous British physician, talked a lot about fever. He gave I think a series of several lectures on fever that have been published in British Medical Journal. And he clearly distinguishes different cases. And one of the cases is the case in which fever, in and of itself, and along with other host responses to infection, is sufficient to eliminate some pathogen. And this happens when we all overcome infectious diseases without taking any antibiotics or things like that. And in Addison's mind, fever was very much a host response that could contribute to overcoming infectious diseases. It's not very clear to me from his phrasing in the second point. He points out that along with fever, we can witness cerebral disturbances. Now, does he mean that this is dysfunction of the central nervous system and it just comes as a price to pay for the beneficial effect of fever? It's not completely clear to me, but at least he seems to promote the idea that fever can be beneficial, and if it is, it may come at some costs for other functions. Osler, American physician, was actually done a lot for the study of medicine in the United States, and is the author of several very influential textbooks through which he has influenced the training and mindset of a lot of students in medicine. And for him, delirium or apathy were clearly symptoms and possible diagnostic criteria for cases of sepsis. So here, once again, in his mind, the behavioral changes are clearly dysfunctional. So I'm not going to go into all of the details of any position that has been taken over history, but I just wanted to point out one thinker and worker in the field that has at least proposed that inflammation is a beneficial response, and that is Metchnikov. In his work at the Pasteur Institute, when he started to work on phagocytosis, he made it very clear that phagocytosis is a beneficial functional response to stop illnesses that are being caused by invading microorganisms. So that is like the prehistory, the setting of the problem that I will be dealing with. And just a little bit more recently, and actually as of the end of the 1970s, people have started to address these issues experimentally. So far, it was very much based on clinical evidence, and of course, you were not going to develop a clinical trial in which physicians would give antipyretics to patients to see if their mortality rate would increase or not. This is only ethical to do in animals up to some point. And interestingly, it has been shown that fever, for example, and trying to inhibit the occurrence of fever does increase mortality rate in animals that were inoculated with bacteria. This has been shown for the very first time in lizards by Matthew Kluger, and has been reproduced for rabbits and mice. So very simple studies. inoculating bacteria to lizards or another organism, and then give to half of the infected animals classic antipyretics such as aspirin. And in those conditions, it was observed that those animals that were inoculated with bacteria and given aspirin showed a higher mortality rate than those animals that were inoculated with the same bacteria but did not get any aspirin. So that did a lot to actually change the way at least some scientists viewed fever, from a potential uncomfortable symptom, it became a potential adaptive host response. So that brings me down to the second part of my talk, in which I will try to transpose the ideas on fever onto the behavioral changes that are typical of being infected with a microorganism. And actually, one can start this question with very much the same approach and same idea as I just showed for fever. The starting point is, if we have a symptom for which we suspect that it may be an adaptive host response, then a natural experimental way to approach this would be to intervene with the symptom, try to block its expression, and then see how the mortality rate of an organism to a certain infection will change. So the Murrays, again at the end of the 1970s, did these kinds of experiments. So their experiment was based on observational data in rabbits, showing that those animals that survived a typical challenge with Listeria bacteria, those animals that survived did actually show a more important weight loss than those that were dying. This is shown here on the left, surviving and dying, and you see the mean loss of body weight in those two groups. So from that, they figured that maybe weight loss, which resulted in part from reduced food intake, was a potential adaptive response to infection. Based on that, they then developed the experiment, which you see summarized in the table on the right, with four different groups. For my purpose, the groups 3 and 4 are the most interesting. So both groups were inoculated with the same concentration of bacteria. Both groups were given ad libitum access to food, except that group 4 was force-fed through an intragastric cannula, whereas group 3 had the same intragastric cannula but was not force-fed through it. So it just had the introduction of that cannula without introducing any food through it. And so if you look at the mean survival time, it's clear that group 4, which was force-fed, shows a lower survival time than the animals that were not force-fed and spontaneously chose to eat less. So this was one of the first experimental evidence that reduced food intake in animals that were exposed to bacterial infection seems to contribute to the survival of the host. This has been reproduced several times, and most recently last year. Now the observational study is on the right, where you see mice being infected with Listeria, again, different doses, and you see their food intake over the days post-infection. So it's a pretty dramatic drop in food intake. Then the experimental intervention study is shown here on the left. So once again, 4 groups, and what was interesting here is that the sort of mean survival in animals that were least manipulated is symbolized by the filled circles. So that line that ends about around a 50% survival. Those are the animals that were giving intraperitoneally phosphate buffer saline injections, and this was the same solution that was giving per os, so through this intragastric cannula. So these animals were receiving non-biologically active molecules. What is interesting, so that would be our control group in this particular experiment. Now, if you look at the lines connected by squares, be that the black squares or the white squares, those are animals that were either given food through the, so force-fed through the cannula, or that were given glucose through that same cannula. And in both cases, you see that the mortality rate increases dramatically. So this very much confirms the previous study from the end of the 1970s. What these authors added, and what I think is interesting, is symbolized by the black triangles. And this is giving deoxyglucose intraperitoneally. So deoxyglucose is a glucose equivalent that cannot be metabolized. So it will block any metabolization of glucose. And that, in and of itself, is sufficient to make 100% of these infected animals survive, suggesting that it's actually glucose that plays an important part in mortality during bacterial infection under these circumstances. So I think these are very strong indications of the potential adaptive nature of reduced food intake in response to at least bacterial infection. For viral infections, the story is different. So we need to be cautious about what we're talking about. This is all related to bacterial infection, and so far has been done mostly with Listeria and Salmonella. So we cannot generalize completely yet, but at least it's interesting. So as I said, as I tried to introduce as an idea, these findings also changed the way, at least in how we in Bordeaux considered behavioral changes during inflammation induced by infection. Instead of it being just mere symptoms, we started to consider them as potential adaptive responses from the host. And this then went along well with a concept that Benjamin Hart developed at the end of the 1980s, in which both fever and reduced food intake and increased sleep were considered to be promoting host survival after infection. And in Benjamin's Hart idea, the behavioral changes were very much to save energy for the fever response to allow to be mounted. Because mounting your body temperature is very costly in terms of energy. So Benjamin Hart reasoned that to be able to do that, one should probably lower other possible expenditure of energy through behavior and moving around. We followed up on all that in Bordeaux in 1997 when I had about arrived there. I was doing my PhD. Another PhD student was trying to characterize more finely with behavioral tests what is actually going on. Because one of the easy explanations for reduced activity and reduced food intake is just to say that animals exposed to bacterial infection experience malaise or pain, and that because of that, or that they are weak, that they show muscular weakness, and that because of that they would not engage in activity or food intake, which is what we typically think to be the case when we, in our medical profession, to a majority thinks that. Now, Arno Aubert, so one of my fellow PhD students back then, he was trained in experimental psychology and developed a pretty nice experiment, I think, in which animals were trained to depend, or half of them were trained to depend on hoarding of food for their food intake. So rodents like to hoard food, so meaning that they will gather food at some place, bring it back to their, in our case, a home cage, but in a natural setting it would be something like a hole or in the ground. And animals do this spontaneously. Rodents do this spontaneously. I mean, we can compare it broadly with us going to the supermarket and buying our groceries and stuff. Before we do not eat at the supermarket. I mean, hopefully we do not eat at the supermarket. So we come back home and then make it dinner and eat it at home in a safe setting. So rodents do the same in a way that they hoard their food from one place, bring it back to their home, and then consume it there. So Arno Aubert figured that this was an interesting sets of behaviors to study, to see whether weakness or pain was the main driver of reduced food intake. So what he did is that he made, as I said, half of the rodents, the rats, dependent on the food they could hoard like a meter away from their home cage. That was the only food they could dispose of to consume. So this is the group that is indicated by no FS. So FS means food supplementation, meaning that the other half of the rats were given the opportunity to hoard for food, but they were always given, at the end of the day, an amount of food as supplementation. So they were supposedly less motivated to hoard for food because they could always count on the experimenter providing them with food at the end of the day. Okay? And then he decided to render half of these animals sick by giving them bacterial fragments, lipopolysaccharide fragments from the gram-negative bacteria, which is symbolized by LPS here, and the other half of the animals were given saline. So on the left, you see ingested food. And so to make a long story short, be that for the food supplemented group or the non-food supplemented group, all animals injected with those bacterial LPS fragments show a reduction in food consumption. Okay? That is what the classical effect was already. Now, interestingly, if you look at hoarding, so the amount of food brought back by the rodent to its cage, then you see interesting differences. So for the group that was not given a food supplement, so the group that completely depended on the hoarded food for what they could eat, still engaged in a pretty important amount of food hoarding. For sure, it was less. So this is what they did before the LPS injection. This is after. So there is a significant reduction in the amount of food they are hoarding. But what is interesting is that they hoard more food than they actually consume. So in spite of them being sick, they still go out to some extent to hoard food and actually bring back more food than they in the end consume being sick. So this, for Arnaud, was a very important point in making the case that sickness behavior is not just the mere expression of the animal being weak or in pain, but there's actually a motivational aspect to it. So the expression of the sickness behavior, in this case reduced food intake or reduced hoarding, depends on the motivation that the animal had here to hoard. And so the expression of this behavior depends on the environment and the internal status of the animal. That here it's a little bit complicated, but it was just to make a point. So if one considered this behavior the expression of a motivational system, then it also makes sense to suppose that there are pathways in the central nervous system mediating these responses. Whereas whether it's just a consequence of pain or weakness, then it would make no sense studying if there were any dedicated pathways, because then we should be studying the pain or the malaise pathways. And so the effect, the phenomenon has been reproduced later on in other rodents and Siberian hamsters. Same thing, when you render these animals sick with the bacterial lipopolysaccharide fragments, they will reduce their food intake, but to a much less important extent, the hoarding of food. So, like previously for the rats, these hamsters will hoard more food than they eat when they are sick. So just a little summary of this concept of sickness behavior. These are 5 ideas relative to the concept. They're not all mutually exclusive, but it gives you an overview of what people have been thinking about sickness behavior. So I talked about Benjamin Hart briefly, who considers sickness behavior as a consequence of the metabolic constraints that come with increasing body temperature. Then my PhD supervisor, Robert Danzer, really made the point that it's the expression of, it's an adaptive response that is the expression of a motivational state. So Arnaud Aubert was also a PhD student of Robert Danzer. And most recently, sickness behavior has been proposed to be also potentially the expression of a pain defense mechanism. But just to be precise relative to what I said a minute ago I entertained the possibility that this changes in behavior after infection may be due to pain. Now, the way that scientists and psychologists have considered pain has also changed meanwhile. Now, pain is also considered the expression of a motivational state. So these last 2 points are not necessarily mutually exclusive. Okay, I won't make it any more complicated than this, and now we'll dive into some good old mechanisms. But I needed to kind of set the scene to hopefully convince you of the idea that the behavioral changes we can see during inflammation are not necessarily a consequence of mere weakness, muscular weakness or pain. They can be considered adaptive in some circumstances, and they can be considered as the expression of a motivational system. And if this is the case, then it makes sense to try and identify, first of all, immune to brain signaling pathways, and in second instance, the central nervous pathways that mediate, for example, the reduction in food intake. And similar things have been done for fever in parallel. So that's what I'm going to do now. I'm going to try and walk you through pretty quickly just to get an idea of what is at stake. I'm not going to spend a lot of time on all the details and all the different hypotheses. I'd just like to expose you to it. If you have questions, feel free. So, as a starting point, our gut contains a lot of microorganisms. We've all heard about the microbiome, and all hear a lot about how good our internal bacteria are for us. This is actually quite a change from, I would say, decades of considering bacteria as really bad for us. So I'm not going to say whether they're good or bad. I think they're probably good at some places, at some distance from our cells. So in the gut, lumen, it's fine. They do good things for us. They make us enjoy food that we could not otherwise enjoy. So great. But as soon as some bacteria either migrate through the epithelial cell barrier of the gut, or if in other circumstances our gut barrier is opened, the epithelial cells are not tightly sealed together anymore because of irritants, or sometimes stress can even cause this, then bacteria present in the lumen of the gut may enter our tissues. And then immune cells will detect bacterial fragments and start to mount local immune responses to the detection of the bacterium and engage in the synthesis of pro-inflammatory cytokines, and other mediators, which are responsible for these classic symptoms of inflammation that I mentioned at the very start. Now, beyond the local players, such as macrophages, dendritic cells, and mast cells, giving rise to the local release of pro-inflammatory cytokines, chemokines, histamines, and prostaglandins. These mediators all have local effects, but some of them also have what we call systemic effects. For example, we know that IL-6, interleukin-6, can act on the liver to promote the synthesis of acute phase responses, phase proteins. And we also know now that some of these cytokines act on the central nervous system to bring about fever. So that is now my background. And so the question really is how, when an organism is exposed to a bacterial infection, the central nervous system is informed and can respond to this by either increasing body temperature or by reducing appetite. What we have used experimentally to study this is, I already mentioned it briefly, is a component of gram-negative bacteria, a cell wall component that is made of lipids and polysaccharides, so hence its name, lipopolysaccharide. You see a schematic chemical structure of it, the lipid part, and then the oligosaccharides part. So these kinds of molecules are common to all gram-negative bacteria. And not surprisingly, macrophages and other innate immune cells recognize lipopolysaccharides via so-called toll-like receptors. So oftentimes in biology, the name of a molecule indicates its function a little bit. For toll, it's not the case at all. It was discovered by a German. And when Germans say great, they say toll. And so that gave the name to the receptor. Maybe that will help you remember it if you didn't know of it. In any case, the detection of bacterial fragments by those toll-like receptors present on macrophages, for example, will give rise to the synthesis of pro-inflammatory cytokines, which then will bring about a local inflammatory response and potentially a systemic inflammatory response. Now, we were interested in how things are being signaled to the central nervous system to reduce food intake, for example. And one of the very interesting findings we came across is that the vagus nerve, so the vagus nerve is this nerve that innervates all of our internal organs, from our lungs, hearts, to all the way down to the colon. And interestingly enough, it was shown that by RT-PCR mRNA coding the toll-like receptor 4 was found in the ganglion of the vagus nerve, which is quite surprising because we were all used to seeing these toll-like receptors being expressed on immune cells, which made sense because they are the cells that are supposed to respond rapidly to the detection of bacterial infection. To see it expressed on nerve cells, or at least in a ganglion of a peripheral nerve, was quite surprising. But we figured that this may be interesting for potentially the vagus nerve could then detect the presence of bacterial fragments, for example, close to the or that may have crossed the gut barrier. And it was already known for quite some time that giving these bacterial fragments intraperitoneally, or also giving a pro-inflammatory cytokine such as interleukin-1 interperitoneally, gave rise to an increase in glutamate release. So glutamate is the main excitatory neurotransmitter in the brain, and its release was increased here in the nucleus of the solitary tract. I mean, don't bother with the name. It's the area of the brainstem where the vagal fibers terminate. So one could suppose that the increase of glutamate in this brainstem structure where vagal terminals can release glutamate had something to do with the detection of bacterial fragments here around the gut by vagal nerve fibers. Okay? So let's say the machinery was there for vagal fibers to detect bacterial fragments and potentially be responsible for the release of glutamate in the brainstem. Now that's all very well, but how does this potentially relate to the reduction in food intake? We know from the literature that the regulation of food intake broadly can be separated in short and long-term regulation. And the idea is that the short-term regulation of food intake, like me now not feeling very hungry after a good risotto, has to do with brainstem circuits. So signals being picked up from the gut, signaling satiety, and entering, in first instance, the brainstem. And glutamate is known to play a role in this phenomenon. So we figured that if we have release of glutamate after detection of bacterial fragments in the brainstem, that this glutamate may be potentially involved in the reduction of food intake. So we decided to do intervention studies with different families of antagonists for the different glutamate receptor types. So to make a long story short, I'm not going to go into much detail. We used, so glutamate receptors come in different shapes and forms and flavors. We played around with broad spectrum antagonists of all of them, but in the end, it was only a metabotropic glutamate receptor antagonist that attenuated the reduction in food intake provoked by bacterial fragments given into the peritoneal cavity. So this is shown here. So we decided to administer that glutamate receptor antagonist into the fourth brain ventricle, which is the ventricle of the brainstem, proximate to the nucleus of the solitary tract. And then we simply measured food intake over 4 hours. So these two bars represent animals that were given saline peripherally, and either a phosphate buffer or the glutamate, the metabotropic glutamate receptor antagonist into the fourth ventricle. So you see that in and of itself, this glutamate receptor antagonist does not significantly alter food intake. Now, when we give the animals our bacterial fragments into peritoneal cavity. You see the drop in food intake, described many times before. But now, when animals are given the metabotropic glutamate receptor antagonist into the fourth ventricle, you see that food intake is somewhat restored. So we think that, in part, the reduction in food intake after inflammation induced by exposure to bacterial fragments is in part due to brainstem action of glutamate on metabotropic receptors. We have gone a little bit into more detail to try and figure out which cells and circuits may be involved, but I'm not going to bother or bore you with that today. So that was one thing, but at the same time, we only partially restored the food intake, which suggested to us that there was more. There may be potentially other pathways. So that brought us back to our office and desk. We sat down and thought about things, and we figured that another way of getting a signal into the brain during inflammation provoked by infection was actually at a level of some small organs in the brain that do not contain a blood-brain barrier. So I don't know if you're all familiar with the idea of a blood-brain barrier. So the blood-brain barrier is made up molecularly by tight junctions, so molecules tightly connected, sealing together brain endothelial cells, which, as a consequence, prevents all hydrophilic, so molecules easily soluble in water, to passively cross the brain endothelium. And this has a lot of evolutionary advantages. This allows, for example, the brain to use amino acids as neurotransmitters, because if there was no such blood-brain barrier, the circulating concentration of amino acids and that concentration just leaking into the brain would make us be in seizure all the time. So the brain uses amino acids as neurotransmitters, but at a much lower concentration than what we have circulating in our blood, especially after a big pasta meal. So that is one intuitive idea of being convinced of the physiological importance of this blood-brain barrier. Another one is that nerve cells use ions to create electric currents. And therefore, they create potential differences across their membranes, which is not something that exists in other parts of the body. So once again, this means that they're actively pumping out some ions. And so if the nervous tissue would simply be exposed to the same ionic composition of the blood, then the nervous tissue could not generate our electric currents that, you know, are happening all the time and firing all the time. So all this just to convince you of the importance of having a blood-brain barrier. Now, if we consider the brain as some kind of master organ that at least would be involved of a lot of things going on in the periphery, there are, of course, the peripheral nerves that can inform the central nervous system of our environment, of our bodily states, but it would also be useful in terms of physiology, in terms of physiological regulation, that the central nervous system could sense what is going on in the blood. And the idea is that that important role is mediated by these areas depicted here in red, called brainstem circumventricular organs, where there is no such blood-brain barrier, so that things can be sensed, circulating molecules can be sensed, right? Now, of course, there's a risk in doing this. If circulating molecules can just spread in these circumventricular organs, then again we would be exposed to the risk of being in seizure all the time. So the story is more complicated than that. There are barrier functions, but around those circumventricular organs. And there is, for example, an epithelial cell layer sealing off those organs from the rest of the parenchyma of the brain. And there are also a lot of immune cells in those circumventricular organs. So the idea is that some of the larger molecules circulating in blood may actually be picked up by immune cells, or phagocytes more in general. And that's an idea then we started to deepen a little bit, because we figured that if in the periphery macrophages were equipped with these toll-like receptors to detect bacterial fragments and give rise to pro-inflammatory cytokine production, if macrophages are present in these brain circumventricular organs, they may express toll-like receptors as well. And this was actually shown in 2001. So TLR4, once again, is this receptor for bacterial lipopolysaccharides. And here you see pictures of the different circumventricular organs showing a positive in situ hybridization signal for the transcript of this receptor. So the receptor is there, so we figured then if we give our animals these bacterial fragments into the peritoneal cavity, and part of that will end up in the circulation, then they may be detected by those phagocytic cells and give rise to local production of pro-inflammatory cytokines in the brain. So that's what we tested. So this is now an example of a circumventricular organ here, area postrema, showing in black histochemical stains for interleukin-1, this pro-inflammatory cytokine, after giving bacterial LPS intraperitoneally. So yes, brain circumventricular organs respond to circulating bacterial fragments by producing locally pro-inflammatory cytokines. Now that's one thing. Does it mean that this interleukin-1 actually plays a role, is biologically active, is released, can act elsewhere in the brain? Because as I said, here there is a barrier, so it may not just passively diffuse into the rest of the brain. So, but our first question was, is locally produced interleukin-1 acting in the brain to, for example, contribute to the reduction of food intake? This is what we did. So nowadays, there are different ways of intervening with the action of interleukin-1. There are soluble neutralizing antibodies. There is a naturally occurring receptor antagonist, which is also used clinically. So we decided to use that, the IL-1 receptor antagonist, also known as anakinra, in the clinic. And we designed a pretty similar experiment in which we gave peripherally saline or bacterial fragments, and then into the lateral brain ventricle solvent or the interleukin-1 receptor antagonist. And this time we looked at, it has been done for food intake, so it also restores the reduction in food intake after LPS. But I just wanted to show you another behavior, which is social interaction. So rats, when they meet a conspecific, they are very much like dogs meeting in the street. You see this very stereotypical social behavior. It's easy to score. And we had already shown in the lab that when you give bacterial fragments, this behavior is severely affected. So if you express it as a percentage of baseline, merely giving those bacterial fragments will almost completely make this behavior disappear. Interestingly, when you give the interleukin-1 receptor antagonist into the lateral brain ventricle, you restore this behavior to some extent. So yes, it seems to be the case that interleukin-1 is acting in the brain to bring about components of sickness behavior. Here it's the reduction in social interaction, but the same thing has been shown for the reduction in food intake. So that was my part on sickness behavior. I hope you are, at least would like to entertain the idea that it may be adaptive. I've tried to show you that there are a couple of ways in which immune signals can be passed on to the central nervous system, be that via detection by peripheral nerves or via detection in the brain circumventricular organs. And then we've seen that either brainstem glutamate or action of interleukin, locally produced interleukin-1, does play a role in bringing about this sickness behavior. Now I would just like to end my talk with the third point, which is more in progress and is not so much what I'm doing myself. It's more I'm observing what is going on, and I'm kind of intrigued. And because this is getting a lot of attention in the so-called high impact factor journals, and also in the press, I would like to just share with you, and maybe we can discuss also a little bit more about this. A first notion on depression as a term. So English is not my native language, so it's always, you probably know that, English is being the international language for science and medicine, and so when you were brought up in a different language, it's always feel kind of handicapped to use English. It's fine, you know, you get by when you're abroad, you order food in a restaurant or a beer, that's fine. And most of the time in science is fine as well, because we have these technical terms that you almost use every day in the lab as well, so most of the time it's fine. But when I start to sit down at the end of an experiment and think of a title for future articles, I was a little bit puzzled by the use of the word depression, depressed, and depressive, and depressed. So what do you do when it's not your native language? Well, you just pick up a dictionary. Or these days we do not pick up dictionaries anymore. They're on the internet. So I thought this was just useful as a starting point to show that all these words, depression, depressed, depressive, and depressed, do have like a normal, like regular language origin before they became clinical terms. And according to the Webster Dictionary, depression is just, or is among other things, a lowering of physical or mental vitality or of a functional activity. Depressed can just mean being below the standard, and depressed is to lessen activity or strength. And just to point this out, there's a lot, I mean, I just did that yesterday. If you type in depression in PubMed, of course you will get a lot of the psychiatric literature. But you also get things like papers by immunologists who show that there was a depressed immune response. So we should be aware of that. And just to make the case, and this is actually inspired what the Webster Dictionary suggested, that they, as an example, they came up with drugs that may depress the appetite. So that's a normal way of expressing oneself in English to describe drugs that may reduce appetite. So I just had a little look, and indeed, in the context of food intake, depression is used a lot. Depression of appetite, depression of appetite, depression of appetite, depressed food intake, etc., etc. So I think you get the point. All these examples have nothing to do with clinical depression as a psychiatric diagnostic category. Now, when it comes to behaviors that are maybe not food intake, I would also like to specify things a little bit. So we can talk, I think, about behavioral depression when we talk about the reduced expression of some behavior, like was the case for food intake before. We can also talk about depressive behaviors, which may be the supposed behavioral expressions and not the affective experience associated with clinical depression. So once again, to make the case that before we talk about clinical depression, there are lots of uses of the word depression that have, I would not say nothing to do with clinical depression, but at least one can adopt a somewhat more liberal use of the word, but one should be very well aware of why one is using it. Okay, all this to get closer to the link that's been made between inflammation and clinical depression this time. So to sum up the concept of sickness behavior, if we use the prototypical experimental paradigm of giving these bacterial lipopolysaccharide fragments, we've seen that it can result in a loss of social interaction, food intake, sexual behavior, and it does increase the time that animals sleep. We've shown that this is in part mediated by the action of interleukin-1 in the brain, and interestingly, some of these effects can be reversed, or at least partially, by giving antidepressants. So that is pretty much the background. Now, we move to clinical depression. Just to last historical slide to show that the link between infection, inflammation, and depression is not new, in spite of what some contemporary authors may say. Some clinicians have at least advanced the idea that infection, especially from the gastrointestinal tract, may give rise to neurasthenia, which was the old term for depression. So it's not a new idea. Now, if we come back to today, I'm going to stick with the DSM-4, the revised version. So you're all aware of the DSM, the Diagnostic and Statistical Manual used in psychiatry. It comes from American psychiatry, but seems to have been adopted by a lot of countries now, not without some resistance, but that's a different story. So why do I put up the 4th revised version, knowing that there is a 5th one? Because I started to work on this when the 4th revised version was still in use. So our starting point was that. And I think with respect to depression, the 5th version hasn't changed that much. But I should probably have a closer look. But anyway, for clinical depression to be diagnosed, according to the DSM-4 revised version, there has to be depressed mood and or loss of interest in normally pleasurable activities for at least 2 weeks. On top of them, that one needs at least 5 of the other symptoms that are listed. So probably are all somewhat familiar with them. Now, if one compares this to the administration of lipopolysaccharides, no longer in animals, right, but in human beings, where it's now allowed as an experimental paradigm, one can actually, a lot of studies have been done actually in humans now, and one can make a comparison between the symptoms of clinical depression and the symptoms that lipopolysaccharide administration induces. And this is just a table that sums up the comparison. So depressed mood, some cases yes, in other cases not after LPS injection. And what is in common is weight loss or decrease in appetite. That is for sure readily observed after the injection of bacterial lipopolysaccharides. But do bear in mind that the initial formulation, according to the DSM-5, is actually weight loss or gain. So lipopolysaccharide only provokes the loss. And the same thing for sleeping. It's either insomnia or sleeping too much. And I know that these criteria seem vague, but that's what we have. It's oftentimes going up or down. Whereas LPS administration only does one thing. And for sure, we do never observe feelings of worthlessness after just giving LPS to human beings. Nor do we observe any thoughts of death or suicide. And if this were the case, of course, it would not be ethical to give this to people. So the comparison, yes, there are a couple of items that are in common between depression symptoms, true clinical depression symptoms, and administration of LPS. But the comparison is certainly not, they do not completely overlap. But there's an interesting subcategory in the DSM revised, the fourth version revised, that is the depression due to a medical condition. And that stipulates mood disturbance with depressed mood that is supposedly due to a general medical condition. And there's a whole list of conditions that may qualify for this. It's cardiovascular conditions, in particular myocardial infarction, gastrointestinal conditions, inflammatory diseases such as rheumatoid arthritis. So this is interesting because this seems much closer to the context of sickness behavior. So indeed, if one goes into the literature and looks at the relative risk of developing depression or depressive symptoms in these different diseases, there is something to say for that. So there is rheumatoid arthritis patients are 2 to 3 times more likely to develop depression than the general population. Asthma, similarly. Interesting for sepsis, severe sepsis, which is supposed to be a typical acute high-grade inflammatory condition. An initial study showed that there was no increased risk. But then a more recent study showed a very spectacular increased risk. So one has to have a close look between the different scales and the different populations. But let's say there's something to be said as well. So I just want to finish with 2 slides showing that cytokines, pro-inflammatory cytokines in depression, for sure there seems to be a case to be made for depression associated with a medical condition. But what about depression that is not associated with a medical condition? There have been studies, now meta-analysis, showing that there is indeed an increase in some cytokines in depression. However, one needs to bear in mind that these increases are, at best, 2 to 3 times increases of 2 or 3 times compared to baseline. Whereas in sepsis, these same cytokines increase by 100 or 1,000-fold. So yes, we're talking about increases, but in depression without a medical condition, it is a 2 to 3-fold increase, and in sepsis, it's about 100 to 1,000-fold. So that's an important point to make. And what happens if one tries to block the action of pro-inflammatory cytokines in depression? For example, by giving a soluble TNF-alpha receptor antagonist. Soluble TNF-alpha receptor. So in a large population of depressive patients, one does not see any overall change in depression severity. However, there was in a subpopulation an important clinical improvement, and that was in those patients that had C-reactive protein levels higher than 5 milligrams per liter. And C-reactive protein is typically used as an inflammatory marker. So maybe, for whatever reason, in a big population, there are people with some inflammatory condition, be that due to a medical condition or not, that may benefit from intervention strategies concerning pro-inflammatory cytokines. And just the latest meta-analysis on this, yes, there are some cytokines that are increased in depression, particular IL-6, interleukin-6, and tumor necrosis factor alpha. And a very recent meta-analysis shows that there is for sure a benefit in anti-cytokine treatments in treating depression associated with medical conditions. So the kind of depression that accompanies rheumatoid arthritis, inflammatory bowel disease, these kinds of things, but not necessarily so far for depression that is not associated with a medical condition. So my conclusion is that pro-inflammatory cytokines mediate sickness behavior. We can call the depressive behaviors if we want to. I don't think it's necessary. Just a mere injection of bacterial lipopolysaccharides is not sufficient to induce clinical depression. And so far, the evidence for the involvement of pro-inflammatory cytokines in depression that is not due to a medical condition is weak. We will see in the future, but so far, this is what I think where the field stands today. But as I said, this is very much ongoing research. So if you're interested, keep an eye out. And we should probably revise our conclusion next time. Thank you very much.